Hello everyone and welcome back to my YouTube channel. So today's topic of the video is most important questions of pharmaceutical biotechnology, that is in your B Pharmacy 6th semester 3rd year. So we have divided this important questions into 5 units. In the first unit, the most important question is biosensors and protein engineering. So biosensors is basically an analytical device that converts chemical energy into electrical energy. And you have to remember the types of biosensors and the components and the few applications. So the components of biosensors majorly include the analyte, that is the substance that you want to detect, which can be either DNA, nanoparticles, antigens, etc. Next is the bioreceptors onto which your analyte will bind, which could be antibody, anything that on which it binds. Next, it consists of a transducer. This transducer is useful for converting this energy. Next, we have an amplifier and a processor. Because the amount of energy obtained is quite less, so we need to amplify it so that we can actually detect the changes and make it more even more sensitive. So these are the components of biosensors. In protein engineering, just remember that there are two types of mutagenesis: site directed and random. Site directed mutagenesis is basically something that is specific to a particular site, but random mutagenesis can occur anywhere. In enzyme immobilization, it is very important question because it has been asked a multiple number of times in examination. There are basically two major methods of enzyme immobilization: reversible and irreversible. In reversible, basically you have attachment and adsorption. In irreversible, we further have cross-linking, covalent bonding, copolymerization, microencapsulation, and entrapment. These are the few methods that you have to remember. And these are done by various reagents. You can say some of the materials required, that is SPVDF, a nylon membrane, any nitrocellulose membrane, gels, SCMC, that is sodium carboxymethyl cellulose, resins, Cefadex and Cefaclor, fibers, and capsules. So these are few of the materials you can say required for individual techniques depending upon the technique we are using. Speaking about the how do actually they work. So basically they work on a single principle. Either you entrap it or you confine or restrict enzyme and it's prevent its mobilization and make it stick to a particular place only. That is what you call enzyme immobilization. Now if you try to represent it in a diagrammatic way, simply remember, suppose if this is a surface, if the substance is getting adsorbed, this is adsorption. If this is a surface, if it's getting bounded through a covalent bond, that is a strong bond, it is covalent bonding. If it's cross-linking, then these materials are cross-linked to each other in a definite web format. If it's microencapsulation, this is a microcapsule, and within that you have your enzyme encapsuled. If it's entrapment, you can entrap it within a lattice like this. So suppose this is your lattice structure, and within this you have your enzymes 1, 2, 3. This is encapsulation, and this is adsorption, covalent bonding, cross-linking, microencapsulation, and entrapment. So these are the few methods that you have to remember. In unit number 2, basically we have cloning vectors. Cloning vectors are agents that are small, circular, extra chromosomal, autonomous, and self-replicating. Whenever you hear a word vector, it means a carrier, something that is used to carry your desired gene, or your design, or your desired material. Cloning vectors are of different types, that is plasmids, they are most commonly used, bacteriophage, cosmids. These cosmids are a combination of the bacteriophage and the plasmids. You have artificial chromosomes, and then you have a phagemid, that is a amalgamation of plasmid and a cosmid. So now in artificial chromosome you have yeast artificial chromosome and bacterial artificial chromosome. Depending upon the type of shuttle vector required, we use the vector. Plasmids are usually capable of 0.1 to 12 kilo base pairs, while bacteriophages have a capability of 10 to 20 bacteriophages. Speaking about the bacteriophage has a capacity of around 10 to 20 base pairs. The cosmids have a base pairing of and capacity of about 35 to 45 base pairs, kilo base pairs. But YAC and BAC can approximately has a capacity of 100 to 1000 or 300 to even 3000 sometimes kilo base pairs. So that is the reason they can replicate into different host cycles, and they are linear when in yeast. But whenever they are in bacteria, obviously they'll be circular because they have to be circular extra chromosomal DNA. So these are few of the types of cloning vectors and the basics of rDNA technology. This unit compulsory and may include 15 marks of your entire excluding your extra questions. This unit is very important and essential to be studied. Now if we go to see rDNA technology, first of all you need molecular scissors or biochemical knives that are called as restriction endonuclease. Secondly, you might need, very important, a desired gene that you need to target. Next you need is a vector, and the most important, a DNA ligase. A DNA ligase basically helps in joining, it helps in attachment. A desired gene is basically something that you target for. A vector can be plasmid, cosmid, bacteriophage. Restriction endonuclease further has three types: type 1, type 2, and type 3. Type 2 is most commonly used and very important because due to its specificity. Next we have is PCR, that is the polymerase chain reaction. PCR is very important because it helps in DNA in vitro amplification. Remember the word in vitro amplification. And PCR has three steps, that is denaturation, renaturation, and polymerization. Each around 1 minute, 2 minutes, 1 minute approximate time. The degree at which it is operated is 95, 55, and 72. You need to remember some terms used, that is the RNA primer. They are primer attachment that is attached via aid of RNA primase that provide the 3' hydroxyl group for complementary sequence. Next, that you need to remember is DNA polymerase. That is very, very vital, DNA polymerase. And you need to have a target cell. So this is the basic of PCR, that is in vitro free amplification. Speaking about the application of rDNA, not that important, but yes, in vaccine and in insulin, it is very important. Insulin is basically derived from pre-proinsulin, which gets converted into proinsulin, which gets converted into insulin finally. Insulin has two chain. It exists as a hexamer, but basically it consists of two chain, A chain and B chain. A chain has 21 amino acids, B chain has 30 amino acids. Together they constitute 51 amino acids. So it is a protein, and via protein engineering, definitely an application of rDNA, we can obtain insulin that is useful in diabetes mellitus type 1 majorly. Next we have is the hybridoma technology. You just need to remember few terms in hybridoma. The HAT medium, hypoxanthine aminopterin tryptamine medium, and it occurs via salvage pathway only. There is no DNA de novo synthesis involved, de novo pathway. Next that you need to remember is the plasma cells that secrete antibody, and the last is the myeloma cells, that is the cancerous cells, and the characteristics is rapid multiplication. So you basically, hybridoma is basically fusion or amalgamation of plasma cells and myeloma cells. Why so? Because antibodies are produced at a quite slower rate, it is not that fast. So myeloma cells rapidly replicate and divide. So when we incorporate these antibodies, they will rapidly replicate and we will get monoclonal antibodies. There are two terms, monoclonal and polyclonal. Basically, it depends upon the epitope. If you want to make it highly specific, we use the term monoclonal, while polyclonal is for a little non-specific because there might be different epitopes as well. But in monoclonal, there is a specific epitope for a specific antibody and antigen. Next is serum and blood serum immune blood derivatives. Blood derivatives could be plasma, they could be cells, and you need to remember the storage. They are mainly stored at around 54 degrees Celsius. That is very vital. MHC, that is the mast cells, must histocompatibility complex. There are two types of MHC complexes, type 1 and type 2. Type 1 has CD8, that is the cytotoxic T cells. Type 2 has CD4, that is the helper cells. Plasma substitutes are not that important, but you need to remember that there are certain plasma volume expanders, either crystalloids, that is sodium chloride, ringer lactate, or some colloids. It could be dextran 40 or 70. It could be degraded gelatin polymer. It could be HES, that is the hydroxyethyl starch, or it could be human serum albumin, that is HSA. There are two terms, bovine serum albumin and human serum albumin. So basically they are, human serum albumin is quite useful. So that's all from unit number 3. Next we'll be having is unit number 4, wherein you have majorly about immunology and you have to study about the immune system. But out of that, more important is the blotting techniques. There are three types of blotting, immunoblotting: southern blotting for DNA, western blotting for protein, and northern blotting for RNA. Just remember, northern has R, RNA has R. So northern blotting in R, southern blotting SD card, you can remember SD card, southern blotting for DNA. So obviously the remaining one would be WhatsApp, that is the Western blotting would be for protein. Basically, there are various steps involved: coating, blocking, washing, visualization. Blocking is essential via bovine serum albumin or skimmed milk because to prevent unnecessary binding of the antibody. And also you can remember the coating could be of polystyrene. Visualization via radio label substances, fluorescence, probes. Visualization can be occurred. Addition of primary antibody and a secondary antibody in case of ELISA, wherein you, even if it's a sandwich ELISA, there's a captured antibody. ELISA are of 4 types: direct, indirect, sandwich. So you have to remember the types of ELISA. Also, next thing that you need to remember is the type of enzymes and the type of substrate. TMB substrate, which turns yellow into blue. Horseradish peroxidase enzyme, beta galactosidase enzyme, alkaline phosphatase enzyme. Do not forget these enzymes. These are very, very, very important. TMB substrate, the beta galactosidase, HRP, that is horseradish peroxidase. Mutation is basically change in the chromosomal or the sequence of the DNA. Mutation could be either frame shift or point mutation. There could be spontaneous, they could be induced, they could be either due to addition, deletion, transversion, replacement or substitution, inversion. So frame shift could be +1, +2, or half or single half, positive half, negative half mutation. Missense, antisense, and nonsense. You have to remember the difference between them. Missense, antisense, or nonsense. And silent. In silent, there is no change in the amino acids. In missense, there is a change in amino acids. In antisense, also there is a change in amino acids. In missense, the amino acid changes is not a termination codon. Here, though, it could not be a termination codon at all. Also, in antisense, it is a termination codon. Missense, it is a little less common. Antisense is quite common. Silent is very, very common. There are various silent mutations. So these are the few differentiating points between the types of mutations. Transposons are basically jumping genes, just remember that. And what are plasmids? Plasmids are vectors. Next you have to remember is the production of B12, that is cyanocobalamin, and penicillin. Penicillin is basically derived from Penicillium notatum and Penicillium chrysogenum. It is a beta-lactam antibiotic and degraded by beta-lactamase enzyme or penicillinase. And it forms penylic acid in acidic medium and penicillic acid in basic medium. In basic medium, it forms penicillinic acid. Just remember this difference. And next is storage collection. How do you store? How do you withdraw? From what person do you withdraw blood? What are the different blood and blood products? Just remember one important concept of adding an anticoagulant in blood. Basically, you add a chelating agent that is called as disodium EDTA. Disodium EDTA. Do not forget this anticoagulant agent. It is very, very, very vital. So that's all for today's video. I hope you enjoyed it, and if you have any doubts, feel free to drop in the comment section. I would be glad to answer them. Thank you so much, everyone.